Hi everyone, I'm Trav. I'm from the Johnson Lab, and we study developmental cell signaling and disease. And we do this in the Drosophila model because we love how easy it is to work with and learn really important things with in relatively quick time. So I think honours is really a great opportunity to sort of put into practice what you've learned over your undergraduate degree and give you a taste of whether or not research is for you. It's not for everyone, but it's a real opportunity to see if it is. I think everyone sort of starts out thinking that research is where they want to end up, and this really provides you with that opportunity. And you also learn a lot. You make some good mates, and you get a taste of what science is really like. So in my lab, I've got a few, I think, are pretty exciting projects underway. One of them is about growth factor trafficking. So we work on cell signaling and receptors and growth factors that activate them. And there's a really interesting mechanism that we don't really understand yet. We know it's interesting because it's really unconventional, where a growth factor gets secreted at the ends of the early embryo. And we're trying to work out how that occurs. It's not understood yet how that occurs, and it has implications for trafficking and secretion of other growth factors, including those that regulate our own development. We're through a project that's been ongoing in the lab. We've found a really interesting mechanism whereby a growth factor controls the number of blood cells there are in a fly. What I like about this system in the fly is that it's really well conserved with humans. Some of the cell signaling molecules involved are very similar. And so what we've discovered is that there's a couple of blood cells that actually control the total population of blood cells, how large that population is. And we're interested in understanding what switches on those growth factor genes in those cells. Another slightly different, a bit more applied project in the lab is looking at rare genetic diseases, rare human diseases. We can model them in Drosophila because over 50 to 75% of human genes are conserved down to flies. And a lot of the ones, particularly involved in disease, are quite highly conserved. And so we're interested in filling the gap in understanding the biology of these genes and their role in disease in order to facilitate treatment developments. And so part of this is in collaboration with Matt Piper's group too. And so in my lab, you really get a taste of modern genetics, sophisticated Drosophila genetics, development, cell signaling, cell biology. You get a taste of human disease. We're always thinking about relevance to humans, as well as some cool techniques like advanced microscopy. And so with all of those sorts of things, I think you end up with a really good training in developmental biology and genetics. And we always look to try and design a project so that it can be published. These aren't the only projects available. Of course, if you have specific interests, we're happy to customize projects around those. All right, well, I hope to see you soon. Good luck. Bye.